Membrane potential refers to the electrical charge difference across the cell membrane when a cell is at rest, meaning it is not actively transmitting signals or involved in any specific activity. At rest, there are more sodium and chloride and calcium ions on the outside, and more potassium ions and large negatively charged proteins on the inside in the cytoplasm. The charge difference between the extracellular material and the cytoplasm interior of the cell, also called the resting membrane potential, is about negative 65 to negative 70 millivolts. We know the term depolarization as it refers to an action potential in muscle cells. It looks like this. But depolarization without an action potential can occur if the threshold is not met. Depolarization describes any change that reduces the charge difference across the plasma membrane, for example, from negative 70 to negative 60 millivolts. Due to the reduction in polarization, the threshold for an action potential is lowered, and therefore this type of change is classified as excitatory. The cell is excited and more likely to generate action potentials. 3 mechanisms cause depolarization: sodium or calcium entry into the cell, or increases in extracellular potassium concentration. Sodium entry into the cell is the most common method. Calcium ion entry into the cell also causes depolarization. Calcium has been found to regulate voltage-gated sodium ion channels, and changes in extracellular calcium concentrations can alter the function of these channels, causing them to open or close. An increase in blood potassium levels, also called hyperkalemia, causes the alteration of diffusion of potassium ions out of the cell and causes a reduction in the membrane potential, depolarizing the membrane and making it easier to reach that threshold potential. On the opposite side, increasing the charge difference between the plasma membrane results in hyperpolarization and an inhibitory signal. Because the membrane potential is increased, for example, from negative 70 to negative 80 millivolts, the cell is further away from that threshold and less likely to generate an action potential. Potassium leaving the cell results in hyperpolarization. This can be through the opening of potassium channels or a decrease in extracellular potassium, called hypokalemia, which encourages more potassium diffusion out of the cell and making it harder to reach that threshold potential. If chloride channels open, negatively charged chloride ions will diffuse into the cell, increasing that membrane potential. Changing the resting membrane potential involves modifying ion concentrations across the plasma membrane. By manipulating these factors, the electrical charge difference across the plasma membrane can be altered.